In this lecture, we're going to look at the electrical activity of neurons. Not all of this is completely clinically relevant, so I want to try to focus on those parts that are. So we're going to look at the different types of signals in neurons, action and graded potentials. Understand a little bit about the ion channels responsible for generating these signals, talk about what's called the resting membrane potential, then talk about the actual graded action potentials, how they propagate, and then how nerves are classified based on their signal conduction. And then most importantly, I think from the pharmacologic angle, is what are some of the agents that are used to disrupt nerve activity via disrupting the action potentials. The resources I mentioned in the previous lectures apply here as well, so you might want to follow along if you need a little bit more background with any standard anatomy and physiology text. Also, there's a lot of really excellent videos on YouTube and elsewhere that kind of go through the whole process of how action potentials are generated. So if something is confusing to you, you might want to go and watch some of those. So as discussed previously, the neurons are electrically excitable and they communicate with one another via two types of electrical signals. Again, we have synapses between neurons, and the majority of those synapses are chemical, so there's neurotransmitters involved as well, and we'll cover all those in a separate lecture. But for the most part, in the dendrites of the neurons, they use graded potentials. This is for short-range communication only. And they're used to initiate action potentials. And then the action potentials will be used for long-range communication. And there's different types. For example, action potentials happen in nerves. That's where we've focused mostly on, in neurons and the axons. But interestingly, muscle can also produce action potentials. So we'll look at those later when we look at myology or study the musculoskeletal system. To generate action potentials requires very special, and graded potentials requires what are called excitable cells. And they are special cells that have very special ion channels in their cell membranes, and that allows them to develop this. Every cell in the body actually has electrical properties. So it's important to differentiate that from what we're talking about here with these two. Every cell has what's called a resting membrane potential. And that is basically a voltage gradient across the cell membrane. So if you look at this diagram here to the right, you see a cell membrane and a plasma membrane. And here's the cytosol, the inside of the cell, and here's the extracellular fluid. Well, cells are able to create a partition between the proteins which are inside the cell, very highly negatively charged, and then potassium, which tends to accumulate inside the cell. And that's partly due to what's called the sodium-potassium pump, which is found in the surface of the cell membranes. Although, interestingly, just based on the properties of water in the cell, and that's a longer discussion, potassium would tend to concentrate inside the cell, and then sodium concentrates outside the cell. And then you have some phosphate inside the cell, some chloride, and so forth. So if you kind of count up and look at all the electrical charge, you find that although cells are in total electrically neutral compared to outside in the extracellular environment, on the very inside of the cell membrane, there's a negative charge. And on the outside of the cell membrane, there's a slight positive charge. So if you were to put a voltmeter in here with one electrode inside the cell and one outside the cell, you would actually find that there's a very tiny voltage, like a battery, across cell membranes. And this voltage is called the resting membrane potential. And that's very important because if cells actually need that voltage to drive a lot of the protein pumps and whatnot that are in the cell membrane. In fact, we can say that one characteristic of death, cell death, is the loss of the resting membrane potential. So all cells, from liver cells, epithelium, everything have this. But neurons have, in addition, other ion gates that are going to allow actually this resting membrane potential to change, to actually discharge and then recharge in an area of membrane. And that's going to allow for an electrical signal to be passed. So that is the generation of the resting membrane potential. All right, before we go further, I need to say a few things about ion channels. So an ion channel basically is a protein which is in the plasma membrane of a cell, and it's going to allow the passage of usually one specific ion. Remember, ions are electrically charged atoms. So cations are positively charged ions and anions are negatively charged atoms. So in an ion gate, basically, it allows, for example, in the picture here, you see chloride is able to go usually from one side of the cell to another. And in really simple ion gates, basically the ions just follow the electrochemical gradient. So chloride will go more towards a positive side because it's an anion, and sodium, being a cation, will go more towards a negative charged area. So that's the basics of an ion channel. So just think of it like a protein tunnel that these ions can go through. Ion gates, a little more specific, these are ion channels that can open or close due to a variety of different factors, and there's a number of different types of ion gates. So muscle and nerve cells in particular have 4 different types of ion gates. So one are called leak channels. Leak channels are the simplest. They basically open and close randomly, and they allow the ion to go through them. And in particular, potassium has a number of leak channels in plasma membranes. So these are important, I should say, because remember that the plasma membrane does not allow ions to go across. So it's not permeable to these things. So you needed a channel to let these ions go back and forth. So leak channels just allow potassium basically to leak out of the cell. And most cells are very leaky to potassium. That plasma membranes have very high permeability to potassium. A second type of ion channel is a ligand-gated ion channel. And basically that opens and closes in response to a ligand. Ligands are substances like a medicinal molecule, a medication, a neurotransmitter, which when it binds to the ion channel, it is able to open the channel, and that allows the ions to go through it. So a good example is the neurotransmitter acetylcholine. That opens specific cation channels, and that allows sodium and calcium to diffuse into a cell and for potassium to leave. So when acetylcholine binds, the channel opens and these ions can redistribute. A third type of ion gate is mechanically gated, and that is true for example, a lot of sensory channels. So for example, channels that are responsive to vibration, touch, pressure, tissue stretching. When those kind of physical things occur, the gate opens and that allows the ions to go through. And that's usually going to cause the nerve to depolarize, and that'll trigger the nerve signal. Finally, and probably most important for this discussion on action potentials and even graded potentials, is voltage gated channels. These open and close due to changes in cell membrane voltage. So if the voltage drops, the channel can open or close depending on what kind of voltage gated channel it is. So if you look at axons, if you zoom in on the cell membrane at a molecular level, you find that they're studded with voltage gated ion channels. And why this is important is that when an area of the membrane depolarizes, that'll open the channels. Sodium's going to move to one side of the membrane. That's going to actually precipitate the signal, and that's going to be carried forward. So we're going to look at that detail as we go here. Okay, jumping back to the idea of the resting membrane potential again. So again, the principle here is that cells have an electrical charge. Usually they're more negative on the very inside of the membrane and more positive on the outside. The total charge, though, again, if you add up all the charges inside of a cell, it's actually the same as outside. So cells are technically electrically neutral, but there's a slight difference along the plasma membrane. And here's a picture of putting a voltmeter in here, and we typically get a measure because the inside is more negative. We usually designate that with a negative sign, and so typical cells range from around 40 to 90 millivolts. The average is around 70, and that's typical of neurons, is between 40 and 90 millivolts. So it's very tiny. A milli is a millionth of a volt. But that's enough to kind of run all the cells, gates and channels and everything else that's needed. So we call this kind of cell that has a resting membrane potential, a polarized cell. So most cells, again, are polarized somewhere in that range. If you kind of look at all body cells like liver cells and epithelium, usually the range is between plus 5 millivolts, meaning the inside is actually more positive than the outside, to minus 100 millivolts. But neurons, for us, we're going to look at 70 millivolts kind of being our target resting membrane potential. Now, I alluded to the kind of things that maybe help maintain the resting membrane potential. One is the unequal distribution of ions both inside and outside the cell. In particular, you have more sodium outside and more potassium on the inside. Both sodium and potassium are positively charged, so that doesn't really affect the electrical conditions. But on the inside of the cell, we have a lot of, and it's depicted in this diagram here by an a lot of anions, and these are primarily proteins or phosphate. So that is going to create a negative charge in general. Because of that, we have that uneven distribution. We have a difference there. Most anions can't leave the cell, and again, that's because they're mostly large proteins, and so they're going to stay inside the cell. And then in particular, there is a pump, the sodium-potassium pump, sodium-potassium ATPase, which moves 3 sodium ions out of the cell and moves 2 potassium ions inside the cell. So the net difference there is we're going to get a little bit more sodium on the outside, and that's going to create that more positive charge on the outside. This does require ATP. And what's very interesting is that biologists calculate that about half of the entire energy of the body, all the food you eat, everything, and all the ATP that's made from that goes into maintaining this pump alone, 50%. So it's kind of crazy when you look at that. And there's been some critics of this idea suggesting actually that doesn't make a lot of sense. It's by looking at water, we can actually find that the water alone is able to make the gradient between potassium and sodium. If you look at sodium, and I think this is a good example of how we often forget about the water in the organism, which is not a good thing. Sodium is actually a smaller atom than potassium. Potassium's one more row down in the periodic table, so it's a larger atom. So if you look at the relative size of sodium, it's actually smaller than potassium. And so you think, well, why would sodium stay on the outside, potassium on the inside? Well, it turns out because sodium's smaller, it has a stronger electrical charge, and it actually is able to attract in several layers of water. Remember that water, we think of water as H2O. It's actually a polar molecule. It has positive charge on the hydrogen end and the negative charge on the oxygen end. And as a result of that, it will line up. Let's say in this case, the sodium is positively charged. The negative charges will line up and form what are called hydration shells around the sodium and so forth. And these shells go out pretty far. If you look at the hydration shells of potassium, they also go out, but not as far as sodium. So interestingly, if you look at the atom with its hydration shells, it turns out sodium is larger than potassium. And as a result, it can't get into the cell. And so basically most of it stays outside, but the potassium can get in and stays on the inside. So just looking at the properties of water actually can explain this electrochemical gradient that happens without the need for a pump. Most conventional physiologists still believe in the pump idea, but again, we might see the evidence kind of turning in favor of this hydration shell idea. Okay, so those are some of the factors that maintain the resting membrane potential. Here's a picture down at the bottom of the supposed sodium-potassium ATPase. So basically ATP is used for energy, and that energy is used to drive out 3 sodium ions, and it's used to bring in 2 potassium ions. And so we maintain a sodium-potassium gradient, so it's said, via that pump. Okay, let's look at graded potentials here first. So basically, graded potential by definition is a small deviation from the resting membrane potential that makes the membrane in this little area either more polarized, and what that means is the inside is more negative, or less polarized, meaning the inside is less negative. So more polarized or hyperpolarized, the inside of the cell is going to be, if here's the cell membrane, if here's the cell membrane, normally we have positive on the outside, negative on the inside. Hyperpolarizing currents actually make more negative on the inside. And then depolarizing currents are going to actually make it more positive on the inside, less negative. So you see that in the picture over here. Here's the minus 70 millivolts resting membrane potential that is standard for neurons. That's the average. So a depolarizing stimulus applied, and remember this is occurring in the dendrites. So if we get a depolarizing stimulus, that resting membrane potential actually goes closer to 0. And so maybe goes up to like minus 60, minus 55 millivolts, versus a hyperpolarizing signal would actually bring it more negative, more down to like minus 80. So we'll see in a moment why that's important. But basically, graded potentials can either create a depolarizing signal or a hyperpolarizing signal. Now, how are these signals made? Well, basically, one of two ways. Either mechanical pressure on the dendrite or the binding of a ligand, and that is typically another neurotransmitter. We mentioned acetylcholine a couple slides ago, but it could be serotonin or dopamine. Those ligand gates are specific, so when the neurotransmitter binds to them, they open, and that allows an ion to come in. Now, it depends on the ion whether or not you're going to have a depolarizing or hyperpolarizing current. So if it's chloride, for example, then what you'll get is chloride rushing into the cell, and that'll make it more negative on the inside. And that's going to be important because basically the more negative you make it on the inside, the less likely that nerve is going to be to fire. So a good example of one of these gates is called the GABA receptor, the GABA-A receptor. So the GABA receptor really is a gate in the dendrite that GABA is a neurotransmitter. It's a variant of a amino acid. So GABA binds to this gate, and it's going to allow chloride to go flooding into the cell, and that's going to create a hyperpolarizing current. And as a result of that, the nerve is going to be less likely to fire an action potential. So basically, it silences the nerve. So that's why we call GABA an inhibitory neurotransmitter. And any drug or substance that also augments the effects of GABA would also inhibit nerves. And a good example of that would be the whole class of drugs known as benzodiazepines, like Valium, Ativan, Xanax, as well as barbiturates. They basically work in different ways. Barbiturates have more of a severe action. But basically, they're going to augment GABA and silence the nerves. And so that's why we find them used less and less, we're seeing now, fortunately, but they're used a lot for anxiety and sometimes sleep disorders. So that's GABA, and that's one example of an inhibitory neurotransmitter. Other ligands might cause sodium to flood in or potassium to flood in, and that would actually depolarize the cell, making it more likely to fire. And a good example there would be the opposite of GABA, which is glutamate. It's a neurotransmitter, which is excitatory. And so we'll look at that when we look at neurotransmitters. So this little area, so again, just to kind of give you context, so here's maybe a dendrite branch. Okay, so the soma's down here, and we got all these dendrites sticking off of it. And then you have this maybe a gate for, in this case, let's say GABA again. And GABA can bind as a ligand to this gate, and that's going to let chloride to flood in. And that's going to cause right here in this area of membrane a hyperpolarizing current. Now, the thing about graded potentials is they're localized. So they're going to stay local and they're going to dissipate with distance. So that's just going to happen in this little area membrane. But imagine there's another GABA channel over here, and GABA binds here from maybe another neuron, and that opens the gate, chloride comes in, we get more negative inside, and that's going to depolarize here. And maybe there's another one here, and that depolarizes. So the interesting thing about graded potentials is that they can be summed together. And that summation is going to reach the soma, and if it goes around the soma and at the level of the action hillock, if it hits a certain area called threshold, so there's a membrane potential that has to be reached to stimulate an action potential. If it reaches threshold, an action potential will be fired. In this case, with the hyperpolarizing currents, it'll actually inhibit that, so it'll prevent the nerve from firing. So the thing about the graded potentials is they are localized, they die out quickly, that's called decremental conduction. They have graded amplitudes. In other words, the stronger the signal, the more the hyperpolarizing or depolarizing stimulus there'll be. And they can be summed together. So remember a dendrite, there can be like 100,000 or more dendrites on a neuron, and they're all receiving input from other neurons, presynaptic neurons, that are dumping neurotransmitters around them, and they're opening up gates. And so all these gates are opening, and all these little currents are being summed together. And then by the time they reach the axon hillock, that might if it reaches that magical threshold number, that might trigger the nerve to fire. So that's the benefit here of action potentials. They can all be summed together like this from multiple ligand signals. So gradient potentials are pretty easy to understand. Action potentials are not that much more difficult, but they have very different properties. So again, these are going to happen only in axons, not in dendrites. And what's going to happen is once an action potential is triggered, it's what's called an all or nothing phenomena. The action potential turns on and then it goes off. It doesn't sum together. It doesn't have a change in amplitude like you see in the gradient potentials. The impulse down the axon with an action potential we sometimes call a nerve impulse. So if you hear that word nerve impulse, it's referring not to gradient potentials but to action potentials. The classic action potential, so this picture at the right is helpful for understanding. This is really a picture of one little area of axon where we've put in electrodes both outside and inside. And we're measuring the resting state. This is again like minus 70 millivolts. So that's rest. What's going to happen in action potential, you're going to see this weird depolarization occur where the membrane potential at that little area actually goes to 0. And then it becomes more positive than the outside. And then it quickly swings back down, usually a little bit under the resting membrane potential, and then back to rest. So that's the classic shape of an action potential. And there are different phases of the action potential. So the phase where basically it's going towards from resting membrane potential minus 70 up to 0 and even beyond, that's called the depolarizing phase. And this occurs because of the rapid opening of voltage-gated sodium channels. So go back to that axon hillock. If normal resting membrane potential is minus 70, let's say, in an average neuron, if the axon hillock, if it reaches threshold potential, and threshold potential usually is around minus 55 millivolts, so a little bit more positive. If all of the signals from the dendrites summed together reach minus 55 millivolts, that will cause voltage-gated sodium channels to open right there. And that will cause basically sodium ions to rush into the cell. And as the sodium ions rush in, they depolarize the membrane, and that's exactly what this upward swing is. So that's the rushing in of sodium ions once those channels open. So that's the depolarizing phase. Then you see the membrane potential drop back down towards the resting, even a little bit more negative than resting, and that's called repolarizing phase. And that happens because of the opening of voltage-gated potassium channels. So the potassium channels are such that they open at about plus 20, plus 30 millivolts. And when they open, potassium is able to rush out of the cell. And that actually is going to repolarize the membrane back to resting membrane potential. So it's complicated. This is happening again at each little segment of the membrane. But let's just start at the axon hillock. Think of the sodium channels opening once they reach threshold. Sodium ions rush into the cell, that depolarizes it to 0 and even more positive than 0. And then the potassium channels open, and now potassium is able to rush out of the cell, and that brings the membrane potential back down towards the resting potential. And then again, there's a little bit of an undershoot where you go a little bit more negative. That's called an after hyperpolarizing phase. And that's basically as these ions are equilibrating and the potassium channels remaining open a little bit too long, they cause a little bit too much of a negative depolarization there. But then it goes back to the rest. So that's the basics of an action potential. You can see that in this picture to the right. So they show you here's a little picture, little part of the axon. Here's an area where an action potential is happening in the blue. Notice the inside is positive, and that's because of the rushing into the sodium ions, and the outside is negative. So it's opposite what the normal axon is at rest, which is shown here in red. Now what's going to happen is this area is going to repolarize. So the potassium channels will open, potassium rushes out, and now the inside the cell becomes negative again, positive on the outside. But now, because of the opening of sodium channels just to the other side, it's going to depolarize that area of membrane. and then that area membrane will repolarize, the action potential will move to the right. So you'll see how, because of the way these channels work, the signal is going to propagate only in one direction, from the soma and the axon hillock down towards the axon terminals. And so that's basically an action potential. So it's very different than a graded potential in that if you look at the action potential, it has a constant amplitude. In other words, you don't get above 30 in this example, millivolts. Unlike a graded potential, the more signal you have, maybe you get more and more negative or positive inside the membrane. Action potential is going to always stay the same. So it's what we call on or off principle. Either the action potential fires or it doesn't. It doesn't like fire more strongly with stronger amplitude. Now the way that nerves actually carry information is to basically code that in the number of action potentials that are fired. So let's say you have a pressure receptor in your thumb. As you press it harder and harder, there's a nerve current that's stimulated from that. And there's an action potential that goes up the sensory nerve, the radial nerve up to the spinal cord. The harder you press on that, the more action potentials will fire. The less you press on it, the fewer action potentials will fire. So we call that's basically what we call frequency modulation, like an FM radio. The more action potentials kind of shows you a stronger stimulus, the less a weaker stimulus. Again, graded potentials are more like AM, amplitude modulation radio, where the stronger the stimulus, the greater the amplitude, and the lesser the stimulus, the lesser the amplitude. So again, a lot of differences between the two. So I mentioned a really important event to stimulate an action potential occurs at the axon hillock, and that is whether or not so-called threshold is reached. And threshold is about minus 55 millivolts in most neurons. Remember, resting is around 70, so minus 55 is a little bit more positive. And different neurons have different thresholds, but basically once threshold is reached, then, and you see that in the diagram here, here's a resting membrane potential, here's a threshold stimulus, here's the threshold voltage. Once that depolarization reaches threshold, then those sodium channels open and we get a depolarization in that part of the membrane. Then the potassium channels open, potassium floods out, little undershoots, and it goes back to resting membrane potential. Notice in this picture you see a couple of threshold voltages or axon hillock voltages that didn't quite make threshold, and so those did not fire in action potential. So that is called the subthreshold stimulus. Again, once the action potential fires, it's always at the same amplitude for a given neuron. Some neurons are plus 40, some are plus 30 millivolts. So information is encoded via frequency, not amplitude. So you see here in one area of axon, they put a little probe in there. Notice the action potentials come through. The stronger the stimulus, the more frequent the action potentials you'll have. And so this is the all or nothing principle. Either the action potential fires, you reach threshold, or it does not. One other important principle here is what's known as the refractory period. And that's the time after an action potential begins, during which another action potential can happen. So basically, the absolute refractory period is, that's shown in red here. If you have an action potential going on, that means that same area of membrane cannot, no matter how strong a threshold stimulus is, cannot be re-stimulated to fire. So nerves basically have a speed limit. They can only fire at a certain rate because of the absolute refractory period. Now there is a relative refractory period, so if you look at this part of the action potential when it goes towards depolarization, hyperpolarization, and then back to normal, if the very strong stimulus comes in again, it can actually reopen those sodium channels, and that can cause another action potential to happen. So we call this the relative refractory period, but it would take a very strong stimulus to get that nerve to fire faster. So if you look at large diameter axons, they have about 1,000 action potentials per second that can occur. In smaller axons in the body, usually there's about 250 action potentials per second, and most neurons average somewhere in between. The relative refractory period, again, is going to be different depending on the different neurons. Now, one final important point here is that because of the refractory periods, action potentials can only propagate in one direction. So as you have one area of membrane depolarizing, it won't back up to the left, for example, in that previous diagram, and depolarize that area that just depolarized because it's still under a relative refractory period. So that means the action potential can only move in one direction. Now there are some tissues like skeletal muscle, which again I mentioned have ion gates just like the neurons, where they have a very low refractory period, and that means the action potential can actually go in both directions across the surface of a muscle cell. But in neurons and axons, they only go one way because of the refractory period. So here's a picture again we saw before of the action potential sort of propagating down the axon. The action potential doesn't die out. What's important is that it's regenerated at each little portion of the axon. So it's not what we call a decremental current like we saw with graded potentials. It keeps its strength as it propagates down the axon, and it's regenerated. So that's why it always is able to keep that same amplitude all the way down. So that's one very important point here about action potentials. Now remember that some neurons, some axons are myelinated. Now in unmyelinated axons, the action potential happens at every little portion of membrane. It moves itself down from the axon hillock all the way down to the axon terminals. But you know, it's going to be kind of slow, and it's going to have to recharge itself along the way. There's another type of conduction. It's called saltatory conduction, and that happens in myelinated neurons, and so that's a little more complicated. What happens is that the action potential is regenerated at each node of Ranvier. Remember, a node of Ranvier is where the myelin doesn't cover the plasma membrane of the axon, the axolemma. So basically the extracellular fluid and the ions, the sodium and potassium, can reach it there. And so the action potential starts at a node of Ranvier, and then it conducts itself, it propagates underneath the myelin on the inside of the axon, and then it's recharged again at the next node of Ranvier. So another action potential happens, propagates down, and it recharges at the node of Ranvier. So that's called saltatory conduction, and that allows for action potentials to travel down myelinated neurons a lot faster than unmyelinated neurons. So we spoke about that in the previous video. So this is a really important kind of concept that allows the nerves to fire more quickly and to maintain those action potentials. It appears like the action potential leaps from one node of Ranvier to another. Again, that's not technically what's happening. There's a ion current that's moving underneath the node of Ranvier, underneath the plasma membrane of the axon, but it basically is the same kind of effect. So the conduction velocity in axons will depend on the amount of myelination. Also, the diameter of the axon, so larger axons conduct faster than smaller axons, and that's because there's more of these channels and whatnot that are present. So large myelinated fibers conduct up to about 100 meters per second, so that's, you know, pretty fast, but compared to small unmyelinated fibers, maybe only conduct about a half a meter per second. So if, for example, again, taking the example of a sensory nerve in your toe, if you stub it, the pressure sensation takes about 0.2 seconds to get from the toe to the brain, versus the pain sensation will take about 4 seconds because that travels along an unmyelinated fiber. And then temperature actually will affect the conduction velocity. So the cooler an area is, a cold limb, that'll actually slow conduction. So if you get really cold extremities, like you're out in the cold, that will actually slow the action potentials. You might notice your muscles, like in the mouth, your jaw, it's difficult to speak sometimes, and that's partly because of the slowing down of the action potentials. So based on those phenomena, we can classify nerve fibers into really 3 basic types: A fibers, B fibers, C fibers in the body. And if we look at A fibers, they're the largest in diameter, about 5 to 20 micrometers in diameter. They are myelinated. They have a very brief absolute refractory period, so they can recharge and another action potential can pass by very quickly. And they can conduct impulses from around 12 to 130 meters per second. That's up to just under 300 miles per hour. These are the axons of most sensory and motor neurons. So A fibers are further divided into alpha fibers and those are present in your proprioceptors, like in the muscles that sense your muscle balance. And these are the conduction velocities over here, so 100 meters per second there. And that's important, as your body in space needs to balance itself. You need very quick updates of information as to where your muscles are. Beta fibers are related to touch and pressure. And then we have the gamma and the delta fibers, and they're related to motor spindles in the muscles. Basically, that's going to let the brain know how much the muscle is stretched. And then the delta fibers on pain, especially cold, and then touch sensations. So those are all examples of A fibers. B fibers are more specialized. They are myelinated, but they're smaller. They have somewhat longer refractory periods than A fibers, and they have speeds up to about 34 miles per hour. And these are typical in sensory nerves in your autonomic nervous system that are sensing your viscera. So for example, in the vagus nerve I mentioned, about 80% of those fibers are afferent or sensory, and those would be examples of B fibers. And then finally, C fibers are the smallest, and these are unmyelinated. They have very long refractory periods comparatively to A fibers, and they conduct at maybe a top speed of about 4 miles per hour. And they carry sensory impulses like pain, touch, pressure, heat, cold, and these are associated with your autonomic nervous system as well. So those are the C fibers. So A, B, C, basically going from larger to smaller, and A and B are myelinated, C is unmyelinated. So one final topic here would be how to disrupt action potentials, and this is used in medicine. So a classic way to disrupt action potentials is to use local anesthetic. Now, what's very interesting about anesthetic is that we don't really understand how it works. We do know it disrupts the sodium channels, and we think what happens is it makes the axon membranes, the axolemma, more fluid, and so it basically these channels can't do their work, so it turns off the nerve. So examples of local anesthetic would be procaine and lidocaine. These are injected in an area, and it basically turns off the sensory nerve activity in that area. So it prevents the action potentials from occurring. More general anesthetics that are used in surgery, there's usually two types. One is to anesthetize your consciousness. The other type is to paralyze. It's a paralytic that's given to paralyze all of your muscle activity, your skeletal muscle activity. So they both have to be administered at the same time. But we think the one that disrupts consciousness, again, works by maybe disrupting microtubules or something in the axons that prevent the normal firing of the action potentials. So that's one example of how to disrupt action potentials, a local anesthetic. Another example is Tetrodotoxin. This is in the pufferfish toxin. It's found in pufferfish, angelfish, some starfish, crabs, worms. It actually is thought to be made by specific bacteria that these fish eat. Basically, it's found in their intestines and liver. So the liver of the pufferfish can concentrate a huge amount of the toxin. And what tetrodotoxin does is it inhibits the voltage-gated sodium channels. So basically shuts them off and they can't depolarize. As a result, then, you would inhibit all of your motor nerves going out to your skeletal muscle. It also inhibits cardiac muscle, but would also inhibit the diaphragm. And so people would basically suffocate. And what is done now, if people get exposed to this, is if they get rushed to the ICU, they're able to be put on a ventilator, and the ventilator assists the breathing until the toxin works its way out of the body. So usually that can happen within just a few hours after ingestion. And if you're unfortunately not close to a ventilator, that usually is fatal. This is actually used in Chinese medicine, or it used to be used. It's recorded in the Shenan Benzaojing as an anticonvulsant. And we can think of that as, a lot of anticonvulsants, as we'll see, actually disrupt sodium channels, usually not as abruptly as the tetrodotoxin. But these would all basically calm down excess nerve activity, which you might see in seizures or in convulsions. So that's one example there is tetrodotoxin. Interestingly, aconitine is an alkaloid in aconite. So there are many different species of aconite, and Chinese medicine uses slightly different species than are used here in Western herbal medicine. But basically, the I'm talking about the raw plant of aconite. The aconitine is an alkaloid which disrupts those voltage-gated sodium channels on neurons, also in the heart muscle and skeletal muscle. So again, same mechanism as tetrodotoxin. This could result in death by respiratory paralysis. So typically the toxicologic signs of this would be you ingest it, you feel numbness and tingling on the tongue. That's kind of the first sign that those sensory nerves are being affected, and then that might eventually result in paralysis of the diaphragm. The treatment of poisoning is actually to administer a little bit of tetrodotoxin or lidocaine or a compound in peony. Interestingly, in Chinese medicine, when fuzi, the processed aconite, is made, it's cooked with ginger and other herbs, and that actually removes large amounts of the aconitine. So it takes out that alkaloid, at the same time preserving some of the other terpenes and other molecules that are found in aconitine. And those molecules have an effect of increasing the activity of mitochondria, especially to make heat, to generate warmth. So curiously, when you process aconite, you're making a totally different medicinal that's found than what is found in the raw drug. So we have to be very clear when we speak about aconite, whether or not we're talking about raw aconite, which can be very toxic, or the processed aconite, which is largely non-toxic. We use it all the time in Chinese herbal medicine, and it has these other beneficial effects. In Taiwan, for example, they use a lot of these Chinese medicinals. Every year there's case reports of several people that either die or they make it to the ICU, put on ventilators because of improperly processed aconite. So that's just maybe just a little call to be very clear where your source of fuzi is coming from, and to be sure it's been processed in the appropriate way. In Western herbal medicine, we actually use very, very low dose raw aconite, especially in homeopathic form. So in homeopathic form, you create a very, very high dilution, usually something like 10 to the minus 6, 10 to the minus 12 dilution, if not more. And this is used for fever, but also anxiety, fear, overactive sympathetic nerve activity. So makes sense in the sense that at those very low doses, you're having a very, very minute paralytic effect on the nerves, which is kind of what you want to do medicinally. So that is aconitine. And one final example of how to disrupt action potentials would be permethrin. It goes by the name of nix or elamite or acticin. Permethrin is used as an insecticide. It's used to treat head lice and scabies. Also pest control in agriculture and the textile timber industries, used to prevent mosquito-borne illnesses by inhibiting it's sprayed on mosquito nets in tropical areas. And this one specifically blocks the sodium channels in arthropods. So the sodium channels in arthropods are different than the ones in mammals and humans. So it doesn't affect us, at least in the same way as it does the arthropods. So it basically causes paralysis in those insects, and they die from that. Usually in humans, it's pretty mild to moderate adverse reactions like skin irritation, maybe some erythema, some itching, pruritus. But it doesn't have the same paralytic effects on the diaphragm and so forth that the other agents I just spoke about do. All right, so that's a little summary of graded and action potentials and how they function, and then some of the things that can disrupt that. In the next lecture, we'll look at neurotransmitters and how specifically different neurotransmitters can create depolarizing or hyperpolarizing currents in the dendrites, and that can either turn nerves on and off. So that'll be in our next lecture.